So in the last few videos, we've talked a lot about DNA, its structure, how DNA is replicated, and even the importance of DNA. But we need to talk about how DNA actually goes from being this instruction manual and blueprint to being a tangible functional protein. And that's the central dogma of DNA. We know that all DNA is replicated from other DNA, right? So DNA gets copied into identical molecules and they can be passed down to future daughter cells and even to future individuals. But how do we get from DNA all the way to a protein? Well, before we can make protein, DNA must first be transcribed into RNA. So all RNA is transcribed from DNA. So that's the first step, taking DNA and transcribing it into DNA's little sister or little brother known as RNA. Now, once we have RNA, we can then translate that message into an actual protein. So you could break it down into a simple little diagram, right? So we've got DNA, and if we want more DNA, all we need to do is DNA replication. Okay. Now, what or how do we get to a protein? Well, that would be the second part of this dogma. From DNA to RNA, that is transcription. So we transcribe RNA from DNA. And then eventually, once we have our RNA molecule, we can finally get down to business and we can translate the RNA into a functional protein. And remember, proteins are made of many amino acids. Okay, so that's the central dogma of DNA right there. Now, let's continue on. What is or how is RNA different from DNA? Well, RNA stands for ribonucleic acid, and the ribo part is referring to ribose, right? So DNA has deoxyribose and RNA has ribose. Now, deoxyribose, as you can see here on the second carbon, is missing an oxygen when compared to ribose, which has the oxygen here on the second carbon. And it's also a nucleic acid. So very much in the same way that DNA is a nucleic acid polymer made of nucleotides, RNA is the same. It is a nucleic acid made of many nucleotides. But the nucleotides of RNA are different. And they're different in a couple different ways. The main difference right here would be the sugar, the ribose sugar, hence the name RNA. But there is another difference here. If you remember with DNA, there were 4 different types of nitrogen bases: adenine, guanine, cytosine, and thymine. However, in RNA, or in the nucleotides that make up RNA polymers, we don't have thymine. Instead, we have a nitrogenous base known as uracil. Now, uracil behaves in the same way that thymine does. So when we talk about complementary base pairs, adenine normally pairs with thymine. But RNA doesn't have thymine. So instead, you would see uracil as the complementary base pair. And cytosine would still pair with guanine. And this shouldn't really surprise you because once you see a picture or an image of uracil, you'll think, okay, that makes sense that it behaves so much like thymine. So let's take a look at that. So we put DNA and RNA side by side. You can see DNA in its double helix. We see 2 strands of nucleotides, right, forming this larger complex DNA molecule, whereas RNA would be a single strand, right, a single strand of nucleotides, and it would still have that sugar phosphate backbone. And in the center here, we have the nucleobases, or the nitrogen bases. Now, if you look here on the outside of the diagram, you can see they both have cytosine, they both have guanine, they both have adenine, and this is where the disparity or difference exists. RNA has uracil and DNA has thymine. And if you look at these 2 molecules, you see there's a lot of similarities here, but there's also some differences. So uracil would be classified as a pyrimidine, okay? So remember, cytosine and thymine are both pyrimidines. Uracil is as well. And if you want a neat way to remember that is you must see the pyramids before your life is over. So UCT. Uracil, cytosine, and thymine, they're all pyrimidines. They're all those single rings, whereas adenine and guanine are the purines. Now, let's get down to business. So we've got DNA, right? And our DNA molecules, right? So we've got ourselves a cell, and inside of the cell we have a nucleus. If we're talking about a eukaryote, and inside of the nucleus we've got our chromatin, right? Our loosely coiled form of DNA. Now, if we were to zoom in on the DNA a little bit, right? And again, this is just kind of a cartoon diagram. We've got our little double helix, our sugar phosphate backbones, and our complementary nitrogen bases here along the inside. And DNA can be almost thought of as like chapters or sections, right? So maybe this particular section of DNA right here codes for a protein. Maybe this section right here codes for a different protein. And this section over here codes for a different protein. And we would call these lengths or these sections of DNA, we would call them genes. So this would be gene number 1, gene number 2, and gene number 3. And each one of these genes might code for a completely different protein, just like you have different recipes in your cookbook, maybe one for cookies, blueberry muffins, and one for a pie. Now, let's say that a particular cell becomes stressed and it needs to make more of this particular protein that is coded for in this gene. So maybe you accidentally slam your hand in the door. I know I've done that before, and your fingernail falls off. That really is not fun. Well, the fingernail is actually made of keratin, which is a protein. And so maybe in the cells of your fingertip, they get squished and they realize, where's the fingernail? It's time to express this gene. It's time to make this protein. And so the cell would go and find this gene number 1, and maybe this gene codes for keratin. And what would happen, let me go back to our blue, is the DNA would be unzipped in that particular section, right? Those hydrogen bonds would be broken. And what we would expose is one of the strands. And this particular strand would have the code. It would be the coding strand. And it would have a series of nitrogen bases, A's and T's and G's and C's. And a little enzyme would attach. And that enzyme is known as RNA polymerase. Hey, that sounds similar to DNA polymerase. What does DNA polymerase do? It reads DNA and synthesizes new strands of DNA. So RNA polymerase would actually read, right, the exposed code, and it would slowly, so it, read along there, and it would slowly produce a single-stranded molecule of mRNA. And the M stands for messenger. And then this messenger RNA molecule, right, would be a complementary copy of the coding gene. And it would be A's, U's, and G's, and C's, right? And the code would be complementary to the coding strand. And eventually the messenger can leave the nucleus and make its way to the cytoplasm, or actually to the ribosomes, which would be the site of protein synthesis. And so think to yourself, what do messengers do? They take information from one place to the next, and that's exactly what mRNA does. And mRNA has a lot of benefits because DNA is really important to the cell, and we want to keep it safe inside the nucleus. And so we can actually transcribe this information. We can transcribe the instructions from DNA into a molecule of messenger RNA, and then the messenger can leave the nucleus and make its way to the ribosome. If something were to happen to this mRNA molecule, there's no harm, no foul, because the gene is still safe inside of the nucleus. Now let me show you a little diagram. And again, I always like to draw things before I show you the actual diagram, so it's not too intimidating. We've got our DNA double helix, and the RNA polymerase has unzipped the gene. And it is slowly reading the template coding strand. And as it reads, it adds the complementary base. So it read T, added adenine, C with G, G with C. And you can see here, when the RNA polymerase read the adenine, it added uracil instead, because remember, RNA does not have thymine. So let's just do a little practice example. So let's say I gave you a strand of mRNA and it had this code. Or no, sorry, what if I gave you a strand of DNA? All right, so here's our little DNA molecule, and this is the code. Maybe it was, let's do T, A, C, G, G, A, okay? And so let's play RNA polymerase. You are RNA polymerase. I am RNA polymerase. What is the complementary mRNA strand? Well, the RNA, I'll write that right here. If we read T, I know that T pairs with A. And then when we get to A, I know A normally pairs with T, but we're talking about RNA, so we're going to have to use uracil. And then C would pair with G, G with C, G with C. And then again, we find A. We want to use thymine, but we have to remember that uracil is unique to RNA, and so we'll plug in another U. And that right there would be a transcribed copy of messenger RNA. That could eventually detach because eventually the RNA polymerase gets to a stopping point. The mRNA is released, and as it says down here, it could make its way to the cytoplasm. And really where it's going is towards the ribosome. Because if you remember from our discussion of cells, we know that the ribosome is the site of protein synthesis, and the mRNA molecule is a message that contains the instructions for the ribosome. In the next video, I'm going to talk about translation. What does the ribosome do with the mRNA molecule when it finally arrives?